Secondary treatment occurs just after primary settling. This stage is essential, particularly for the removal of organic matter. What does secondary treatment consist of? What processes are used? We will look at both fixed culture and free culture processes. The purpose of secondary treatment is to remove biodegradable organic matter containing carbon or nitrogen contained within wastewater. In certain cases, it can also allow a portion of phosphorus pollution to be treated too. Without treatment, the significant presence of this organic matter in the natural environment would facilitate the proliferation of microorganisms. In order to degrade this organic matter, microorganisms would then monopolize the oxygen present in the water, which would deprive the aquatic environment of this resource essential to life. The secondary treatment, therefore, uses microorganisms to degrade the pollution, but within the station. All of these microorganisms are called biomass, or purifying biomass. This is why secondary treatment is described as being a biological one. There are two types of processes in secondary treatment: anaerobics, biological processes, and aerobics, biological processes. Anaerobic processes are carried out in the absence of oxygen in a molecular form or in the combined form with another chemical element. These are not often used in wastewater treatment processes unless phosphorus is going to be treated in a secondary treatment process. They will be encountered more frequently in sludge treatment processes or in industrial effluent treatment processes. Now let us look at aerobic biological processes. Aerobic biological processes take place in the presence of oxygen. There are both free culture and fixed culture processes. For free culture processes, purifying biomass is suspended in the water to be treated. Here, we will only look at intensive free culture, that is activated sludge. For activated sludge, the development of the purifying biomass that evolves freely in a tank is maintained. This tank is fitted with a stirrer which keeps the culture in suspension. It is also fitted with an aeration system which enables it to maintain a sufficient level of oxygen essential for the development and the activity of microorganisms in the biomass. Different types of activated sludge tanks exist. These are a simple aeration tank, a tank with a zone in anoxia and an aerated zone, and a tank with an oxidation channel. A zone in anoxia is a zone where there is no molecular oxygen. In fixed culture processes, purifying biomass is fixed to a supporting medium via a biofilm. A biofilm is a structured cluster of bacterial cells attached to a surface. Amongst these processes, bacterial beds, biological discs, or more recently, biofilters are found. In the case of a bacterial bed, wastewater that has been through the settling process beforehand trickles into the mass of porous materials or with halos serving as a purifying biomass supporting medium. The purifying material is sprinkled by the water to be treated using rotary distributors called sprinklers. Biological discs are a process also based on the contact or fixed biomass with the water to be treated. Through a rotating movement, semi-immersed discs allow the biofilm present on their surface to be in contact with the water to be purified. The discs are semi-immersed in order to allow the bacteria that break down organic matter and that are present to be oxygenated on a disc. Biofilters are based on the use of a filtering medium which can be granular. This filtering medium is colonized by purifying biomass and through the transit of the effluent to be treated. What you need to remember. The purpose of secondary treatment is to remove biodegradable organic matter using microorganisms present in the natural environment. Aerobic and anaerobic secondary treatment processes exist. Amongst aerobic biological processes, free cultures and fixed cultures exist.